And the second speaker is Dr. Tasuku Hirayama. And he has a very wonderful background, but I just want to emphasize here, he developed a raw NX turn-on fluorescence probe for imaging-labeled iron plus 2 plus iron, and driver is to expand the repertoire of iron to selective fluorescence. And some of the probes he made has become a standard for imaging label iron 2, which plays an important role in ferroptosis. And then the lab, like us, and then we use many fluorescence probes from his achievement. Thank you very much, and welcome. Please, now.
Thank you for your kind introduction. First of all, I thank the President Ueno and the staffs from Dojindo for their kind invitation and hospitality. It is my great honor to have a presentation in this memorial first Dojindo forum in China. And my research interest is to develop chemical tools for studying ion biology. So my talk would be directed much more chemistry side. Today, I would like to talk about 3 topics. The first one is Fluorescent probe for ray vital ion, Fe2+, from fundamental aspects to their applications. The second topic is Heme selective fluorescent probe, and the third one is about the fluorescent probe can work as Ferroptosis inhibitors too, which we working on recently. Please let me talk about the introduction of ion cellular biology. The ion is the most abundant transition metal in our body, and most of ion exists as protein-bound form, and at the same time, a small portion of ion also exists as protein-free or weakly protein-bound form. They are called as ray vital ion. And the major component of ray vital ion in living cells should be Fe2+ rather than Fe3+ because of water solubility and radioactive cellular environment. And much abundant glutathione stabilizes Fe2+ rather than Fe3+ in living cells. And transporters chaperones involved in intracellular ion homeostasis recognize Fe2+ rather than Fe3+ as their substrate, indicating that the Fe2+ is much more available form than Fe3+. So to study the ion homeostasis, physiological aspects of ion homeostasis, Fe2+ must be the target. At the same time, Fe2+ is highly relevant to oxidative stress. The Fe2+ react with hydrogen peroxide to generate hydroxyl radical, and if Fe2+ react with lipid hydroperoxide, it will generate a lipid peroxide radical. And overproduction of these chemical species causes cell damage and ferroptosis. So both for the physiological aspects and pathological aspects, we need to know the behavior of Fe2+. So that is why we started to develop a new chemical tools for the study Fe2+ ion. When we started, only 5 examples of ion selective fluorescent probes have been reported. Although ion zinc specific fluorescent probes, there are many zinc specific fluorescent probes. The calcium was the only commercially available fluorescent probe to detect Fe2+ and Fe3+. And this one is not selective to Fe2+ and Fe3+, and other metal cations only shows also shows a turn off response. Then calcium cannot avoid a turn off response. The chelation-based strategy is a general design strategy for the metal cation fluorescence sensing probe. And fluorophore is tethered to chelation moiety. And when the chelation moiety binds to metal cation, the fluorescent property is changed. and we can see the fluorescence change upon binding to the metal cation. However, in the case of Fe2 plus or Fe3 plus, the fluorescence response must be turned off because Fe2, both of Fe2 plus and Fe3 plus are strong fluorescence quencher. So we needed to change the design strategy to obtain turn-on type of fluorescence probe. We decided to use reaction-based strategy. In this case, fluorescence quenching domain is tethered to fluorophore to quench the fluorescence. When this probe reacts with the target molecules, the quenching domain is clipped off, and we can see turn-on response. In this case, we targeted Fe2 plus, and what chemical reaction can be used for the selective detection of Fe2 plus is the first question. We found one of the answers in the protective group's handbook. Because I'm an organic chemist, I think for almost all organic chemists, this handbook is like a Bible. And I found this chemical reaction in this handbook, and this chemistry is used for the protection of tertiary amine. And the deprotection of this compound can be done by a mild reductive deoxygenation reaction using iron. So we expect that this reaction can discriminate Fe2 plus from Fe3 plus because Fe2 plus is more reductive than Fe3 plus. How to incorporate this chemical reaction as a fluorescence turn-on switch is the next question. We chose rhodamine B as the fluorescent scaffold because this one contains tertiary amine here, and this is a strongly fluorescent compound because its strong fluorescence is due to the localization of this electron pair. When this tertiary amine is converted to N oxide, the electron pair is used for making a bond. So in this case, this electron cannot be incorporated into this conjugation system, and nitrogen atom is separated from the fluorescent structure. So this causes attenuation of its fluorescence. When N oxide reacts with Fe2 plus, and deoxygenation occurs, this compound should be going back to the highly fluorescent compound, and we can see turn-on response. That is the mechanism of we expected. This is the first example of our N oxide-based fluorescent probe for the selective detection of Fe2 plus. This one is rhodamine N oxide. That's why we call this RONOX1. And this one shows only a weak fluorescence. And after the reaction with Fe2 plus, this one was converted to highly fluorescent compound, and we can see turn-on response. Here shows the fluorescence spectral change. Upon the addition of Fe2 plus, we could observe the turn-on response. And the most characteristic point of this chemistry is its high metal selectivity. Only Fe2 plus triggers fluorescence turn-on, and other metal cations did not show any fluorescence increase. And this one can be applicable to live cell imaging application. And I was lucky to have a good collaborator, Professor Shinya Toyokuni from in Nagoya University. He was supposed to attend this meeting, this forum, but unfortunately he is not able to be present because of sickness. I'm very sorry. But anyway, he was the first person showing interest in our fluorescent probe because his research interest is about the ion-induced carcinogenesis. And his hypothesis is that Fenton reaction active Fe2 plus must be accumulated in the carcinogenesis site. So he wanted to discriminate Fe2 plus from other ion species. So our fluorescent probe perfectly satisfy his requirement. So he applied a first probe to his ion-induced carcinogenesis model, and he revealed that Fe2 plus is accumulating in a carcinogenesis site. After his success, I received many offers, many inquiries from researchers, not only from Japan, but also from all over the world. And now many collaborative work have been published. And now some of our first probe is commercially available from Dojindo Chemicals. And here shows a history and the list the first probes we published so far. And I would like to talk about some examples using some of these fluorescent probes regarding the ferroptosis study. The first one is multicolor imaging application of Fe2 plus imaging using organoid-specific fluorescent probes. We have several types of fluorescent probes, and rhizosome, ER, and mitochondrial-specific fluorescent probes were used at the same time. And it is important that these three fluorescent probes have similar responsiveness, but different colors and different targeted organelles. And one of these mitochondrial targeted one is commercially available from Dojindo. And anyway, I applied a cocktail of these fluorescent probes at the same time to monitor ferrous ion status at their targeted organelles. Here shows just the demonstration, and Fe2 plus treated cells showed significantly higher fluorescence signal, and the distribution patterns are specific to each targeted organelles. Here shows the mitochondria and the rhizosome and the ER. So all of these fluorescent probes showed fluorescence signal, fluorescence response in their targeted organelles. Then I would like to skip the ferroptosis introduction. But anyway, although this is ion-dependent cell death, but the ray-viral ion status remained unknown. So our interest is to know the ray-viral ion status. I mean the Fe2 plus status, alteration of the Fe2 plus status during the ferroptosis. And here shows the organelle-specific analysis of ray-viral ion status by using the cocktail of the mitochondria, ER, rhizosome-specific fluorescent probes. And we observed significant fluorescence increase in the elastin-treated cells, but the mitochondrial fluorescence signal increase was independent of the ray-viral ion. Another control experiment supposed that in the elastin-treated cells, the fluorescence overaccumulation was observed. So the DFO cannot rescue the DFO must rescue the cells from ferroptosis, but the fluorescence signal was not suppressed by the addition of DFO. So this fluorescence increase, we concluded that this fluorescence increase was independent of the ray-viral ion status. But in contrast, the rhizosome and ER fluorescence signal was completely suppressed in the presence of DFO. So the rhizosome and ER ray-viral ion concentration must be upregulated upon the addition of elastin. So here shows the summary slide, and elastin induces upregulation Fe2 plus and in ER and the rhizosome, but mitochondrial fluorescence signal was independent of Fe2 plus status, and how Fe2 plus changes during the elastin-induced cell death remains unknown. But anyway, the Fe2 plus upregulation is essential for the lethality in the ferroptotic cell death. And the next topic is about the high throughput screening system using extremely sensitive fluorescent probe for ferrous ion. And as I told, ion is the most abundant transition metal species in our body, and 4 to 5 grams of ion is contained in human adult. And iron deficiency causes anemia, dysmotility. All of them is caused by the dysfunction in the energy production systems. And also iron overload causes severe diseases such as cancer and neurodegeneration. Iron deficiency, for the therapeutic treatment of iron deficiency, iron supplementation is often used. And in the case of iron overload, chelators are used for the treatment. And the contents of iron supplies, iron salt, such as ferric citrate. And these treatments are direct and simple, but sometimes they are too aggressive. Iron supplementation often causes iron overload, and chelators may sometimes causes iron deficiency. This situation is similar in the cell biology field. We use iron salt or chelator to induce iron overload and iron deficiency, respectively. So to study iron biology more deeply, we need a new compound that can regulate iron homeostasis without addition of iron salt and chelator. So the compound that can stimulate ion transport or ion homeostasis, ion equilibrium system in cells must be a good drug candidate to treat ion deficiency or ion overload. So there are several compounds have been reported. Iron-mycin, hinoctiol. These two compounds can regulate cellular ion status without adding ion. So, motivated by this success, we started a new drug discovery project by using high-throughput screening of chemical library. For the high-throughput screening of chemical library, we need a robust and highly sensitive fluorescent probes. The enhancement of the sensitivity is the first hurdle to clear. To enhance the sensitivity, we knew that the cyclization of N-oxide domain here to like this compound can enhance the sensitivity, and we designed and synthesized new 7 compounds having cyclic amine N-oxide platform, RONOX2 to RONOX8, and evaluated their responsiveness to ferrous ion. And all of the new compounds showed better response than the first generation Prof, and RONOX4, 7, and 8 showed extremely higher response and over 100-fold increase within 10 minutes. However, unfortunately, only RONOX4 worked in living cell application, but RONOX4 showed significantly better response than RONOX1. A pair of these images were obtained after contrast adjustment, but in the case of RONOX4, this is also raw images. So if you have worked the microscopic analysis, you can understand how sensitive this fluorescent probe is. And we applied this probe to monitor the labile ion status by several artificial stimulations. Copper did not affect the labile ion status, but zinc and ascorbate upregulated Fe2+, and hydrogen peroxide and nitric oxide downregulated the Fe2+, which are easily visualized by RONOX4. Encouraged by these results, we established a new high-throughput screening system by using RONOX4, and 3,400 compounds were screened by a high-content screening system with 96 well-plate platform. During this high-throughput screening, we observed not only fluorescence increase, but also fluorescence decrease. This indicates that our high-throughput screening system can discover not only the ion up regulator, but also down regulator of labile ion. And we repeated 3 times and found romofangin as a hit compound that can upregulate ray ion as low as 5 micromolar concentration. Then the next question is how romofangin upregulate the ray ion in cells, and we this is now ongoing project, but we found that total ion was not changed upon the addition of romofangin. This means the transporting system was not affected by romofangin. And we checked several proteins involved in the ion homeostasis, and ferritin degradation was observed. And ferroprotein is an exporter of ion. A transferrin receptor and IRP2 were also examined, but they were not changed at all. It is reasonable that ferritin degradation upregulates Fe2 plus level because thousands of ions are contained in one ferritin molecules. And when ferritin is degraded, the ferric ion is released from ferritin molecules, and these Fe3 plus is readily reduced to Fe2 plus by the abundant glutathione. So that is why Fe2 plus is upregulated. The next question is how ferritin is downregulated upon the addition of romofangin. The lysosomal degradation is the major pathway of the ferritin degradation, but lysosome peptidase inhibitors did not affect the upregulation of Fe2 plus. So further experiments are required to reveal the whole mechanism of romofangin, but unfortunately, commercial source of romofangin has been stopped. So, and I have no choice but started total synthesis of romofangin, and finally we obtained the romofangin from the commercially available compound after slugging for 4 years, and hopefully I will present the whole mechanism of romofangin in near future. So we established new high throughput screening compatible fluorescent probe, and this is Ronox 4, and this is ferro orange. Maybe some of you may use this one. And we found romofangin as a new compound that can degrade ferritin in cells and upregulate Fe2 plus ion. The next topic is heme selective fluorescent probe. And as you may know, Heme is a metal complex of iron and protoporphyrin 9 and plays many important roles, essential roles as cofactors of heme proteins, such as P450, hemoglobin, and electron transporting change. And the situation is similar to ray ion, and protein-free or weakly protein-bound heme also exists in living cells, and they work as cell signaling molecules. Here shows one example. BAC1 is a transcription factor that is activated upon the heme binding. So that is why heme acts as a cell signaling molecule. And also heme is involved in oxidative stress due to its high redox activity and lipophilic properties. The intracellular heme trafficking remains unknown. Here shows a figure cited from this review, and many question marks. You can see many question marks alongside arrows, and less is known about heme trafficking than ray ion. So we wanted to know the trafficking of heme by making a new chemical tools for ray heme. So, and I expected that our N-oxide chemistry can be used for the detection of ray heme because the metal center of ray heme also Fe2 plus. However, we must overcome the difference in the intracellular concentration range because ray ion is 10 to 100-fold higher concentration than that of ray heme. So we need to tune the reactivity of N-oxide to have less reactivity to ray ion, and higher reactivity to rival heme. So to make a long story short, we found difluoropiperidine N oxide with Rodol scaffold can achieve the heme selective fluorescence response. Here shows the results, and 1 micromolar hemine induced over 200-fold increase within 10 minutes. In contrast, Fe2 plus showed only a 2 or 3-fold increase even after incubation for 1 hour. So this means H Humlox is completely selective to heme over Fe2 plus. And then living cell live cell imaging application is shown here. And we use 5-amino-levulinic acid to induce heme biosynthesis, activate the heme biosynthesis, and we observed process increase in the amino-levulinic acid treated cell. But in the case of co-treatment of succinoloston, which is an inhibitor of heme biosynthesis, the fluorescence increase was completely suppressed. And we further applied the monitoring rival heme level by using zinc protoporphyrin 9, which is an inhibitor of heme oxidase. Heme oxidase is enzyme of heme degradation. And we observed higher fluorescence signal than the amino-levulinic acid treated only amino-levulinic acid treated cells, and we conclude that our fluorescence probe can detect rival heme selectively in living cells. Then here shows one example to monitor the rival heme level induced by nitric oxide. And heme binding proteins can release heme from its binding pocket upon the addition of nitric oxide, because nitric oxide can nitrogyrate cysteine residues nearby the heme, and when cysteine residues is nitrogyrated, the affinity to heme reduced, and heme is released from the binding pockets. And we checked, we applied to see that this happens in living cells or not. We observed fluorescence increase by the addition of nitric oxide donor, NOC5. This is the only example that the rival heme release without heme exogenous source of heme addition. The other application is to monitor another application of our heme fluorescence probe is monitoring rival heme and rival ion during the ferroptosis at the same time. We have heme selective fluorescence probe, this one, and we have also Fe2 plus selective fluorescence probe. This is inert against heme. And this was green color fluorescence probe, and this is a red color fluorescence probe. So this fluorescence probe has a different color. So these fluorescence probe can be used at the same time. Then elastin-induced cell elastin was added to cells, and the cells were stained by our pair of fluorescence probe, and we observed fluorescence increase in the elastin-treated cells for the Fe2 plus selective cells, as I showed before, and DFO suppressed the fluorescence increase in the Fe2 plus selective fluorescence probe channel. In contrast, elastin-induced cells showed upregulation of rival heme, but DFO did not affect its fluorescence. It is because that DFO cannot curate heme itself. So, and we need further experiment in this case too. But rival heme is upregulated during ferroptosis, but the rival heme itself is not directly involved in the lethality, because DFO cannot down-regulate rival heme level. And why rival heme is upregulated remains unknown, so we need a further experiment to study this phenomenon. But anyway, this is the first example of rival heme upregulation during the ferroptosis. to wrap up my talk, I would like to talk about the future of ion bioscience. And Roger Chen developed Nobel Prize as a GFP staff, and he developed the calcium indicator FURA2. And after he developed FURA2, the calcium biology papers rapidly increase. So how about the case of ion biology? We have reported many types of ferrous ion Fe2 plus selective for the probe, and there are a number of papers. Oh, what is? Oh, okay. Anyway, yeah. Anyway, I continue. Okay, a number of papers referring to probes keep increasing. So I hope the number of papers keep increasing and increase, and finally, a new journal dealing with cell ion biology will be published in the near future. Okay, I'd like to summarize my talk by showing a history of across the probe again. And we have developed new chemical tools to monitor the not only labile ion, but also labile heme. And some of them are commercially available from Dojindo, and some of them are commercially unavailable. So if you need this one and the other fluorescent probe, and heme fluorescent probe is not commercially available. So if you need these fluorescent probes, please ask me. And I would like to thank all the members who contributed to this work and financial support. Thank you for your kind attention. I'm happy to take any questions.
Yeah, thank you very much. And then this is really for the iron science, and then we need these probes for all the iron research.
Thank you.
Not only for the ferroptosis and also for pure iron metabolism.
Yeah, right.
And then actually there's quite a few questions here, and so for me, I think the probe you use and designed in principle is an iron chelator.
Not ion chelator. Yeah.
So because they first need to bind an iron, right?
Yes, bind not
the binding, but of course it's the they have a bi function. First they bind iron, and then next they make the fluorescence like bright this, right? And then for me, in my case, I'm an iron metabolism person, and then the iron needed homeostasis.
Right.
When the probe binding to the iron is very critical. Actually, you already show it because it's very critical for the affinity. Then the affinity is high. It's not really good because they need this homeostasis. If you create more, and then the iron 3 just compare. Iron 2 and 3, they just if the one side is less, and then from another side go to this side, and then
exactly.
And then so in your case, how do you balance it? You have really highly sensitive to the cellular neighbor iron pool, but you won't change the cellular really the iron homeostasis disruption. Yeah, this please comment.
Okay. It depends on how to use our fluorescent probe. It depends on concentration and incubation time and the incubation conditions. So for imaging study, we use a very short time incubation time and low concentration, only 1 micromolar concentration with 30 minutes incubation. That does not alter the ion homeostasis because the Fe2 plus reacts with our fluorescent probe, and Fe3 plus is generated, but Fe3 plus is quickly resist to Fe2 plus. And the reductant is glutathione, this is much abundant than the probe concentration. So the homeostasis must not be affected by the fluorescent probe.
But for in the last step, you use your probe and then you observe this is a ferroptosis inhibitor.
Yeah, right.
And then this is really just cleared the iron. But this is the cleared iron and then make the cell survive.
No, our fluorescent probe doesn't have curating motif and curating property. So just oxidize Fe2 plus to Fe3 plus.
Okay. And then make the
It's
the
equilibrium.
I3 alert is not active anymore. Okay,
Right. Yeah.
Okay. And also there are some online questions. Yeah, this is also I think is my interested, and we have some work with Gorge, and then we hope you can develop the Gorge probe for us.
Okay. Actually, we have developed and also reported goals specific for the probe.
Okay. And then also in Dojindo, we have this available in Dojindo.
No.
Oh, not yet.
Please ask them. Was negotiating.
And actually, there's some question. Let me see. Can you comment on it? And I think it's not really relevant to your talk. The FUR1, the ferroptosis inhibitor FUR1 affect expression of XCT transporter protein?
Which is not question for my talk.
Yeah, right. Let me see. So there's one question. Ferroptosis and oxidative stress seem very similar. And is the key difference between them? Maybe here this is for additional to iron accumulation, I think. So can you come on it?
Okay.
So you want to come ferroptosis and oxidative stress. So actually, I think in another way means does oxidative stress cause ferroptosis always, or just in a very specific case?
In my opinion, oxidative stress is complicatedly relevant to the ferroptosis. I cannot answer. I don't have an exact answer for that.
In my opinion, seems not always. Also depends on the context.
Right.
Yeah.
Not always. Yeah. Exactly.
And then there's another one for the timing. The timing for detecting this indicator of ferroptosis after treated cells with uracil. For timing, if the timing is the same, for example, after 24 hours of treatment, or only maybe the partial cell death, is there any undefined and appropriate treatment time can be make the indicator show or the ideal or trend?
That's a good point. We explored time course experiments by elastin-treated cell death, and our fluorescence images were taken just before the ferroptotic cell death, because the cell death makes the probes accumulation upregulated. So we checked the cell viability, and images were taken before cell death happens. So in this case, we observed ion upregulation after 3 hours, 5 hours incubation for incubation of elastin. So after that, cells started to die. So the images
It's not accurate to
yeah.
present the iron concentration.
Right.
So
First, my lab also used the ferro orange and the metal ferro orange, and we actually observed that maybe during the early phase or of ferroptosis, or the cell is still okay without any sign of death. And we observed that mitochondria actually can kind of observe cellular ion, meaning the ferro-orange signal getting very high, that's the early stage, and then the signal getting reduced, and right before the onset of ferroptosis, we think maybe mitochondria could function as a sink for liable ions in the early phase. There may be a way to protect the cell from death. But when they accumulate too much then they're causing damage to mitochondrial membrane, and through Fenton reaction and the onset of ferroptosis.
Thank you for the comment.
We have actually published a paper in Cell Discovery using your prop, and
thank you for the comment.
Okay. And then the next question is whether it is possible for the scientists to measure the iron and heme simultaneously.
It's possible. Yeah.
So the answer is possible.
Answer is possible. Yeah. This is an example. Yeah. Fe2 plus and Heme can be orthogonally detected by a pair of these fluorescent probes. Yeah.
So I normally for us the iron field, the heme, and also as you say, and iron also binding by quite a different small molecules in the cells. It's not really free iron. So this is just called the neighbor iron core. And then for the heme, it's the same. And then you use the probe to detect actually is it free heme, or it's very similar like the neighbor heme pore?
Yeah, it's a good question, and we expect that rival heme pool, because our fluorescent probe can react with weakly bound heme. So in the presence of some heme proteins such as cytochrome C and hemoglobin, may heme in the hemoglobin or cytochrome C can react with our fluorescent probe weakly. So that is why we think that weakly protein bound heme and maybe protein free heme is detectable by our fluorescent probe.
Okay, and then in the slide, and then because you use uracil, and then uracil generally we think take to induce the ferroptosis, right? And then this is the more heme, perhaps because the cells were going to be die, and then some iron binding protein just release it.
Right.
And then make your heme bright. You hear this is a high heme fluorescence signal.
Yeah.
This just come on here.
Okay, yeah, yeah.
Mhm.
But I have no idea why heme is upregulated.
Yeah, it's not
this-
heme upregulated
Yeah.
because the cells are going to die. The heme binding protein, they won't binding so tightly.
Yeah, right.
And then it's possible after you resting and then the heme normally I just grab it and then after resting I'm and then just release it.
Yeah.
And then make your heme signal stronger.
Yeah.
I'm not very sure, just guess.
Okay. Thank you.
And then there's some more question. Let me see. Can this detection probe directly enter the mitochondria, lysosome, or ER? And what's the principle of high affinity for iron 2 without binding to iron 3?
Oh.
So
About the
I think
first question, and
yes.
organ targeting is the nature of the fluorescent probe by themselves. Well, yeah, so they directly go into their targeted organelles.
Yeah. So the first answer is they can go directly to the organelle. For instance, the mitochondria, lysosome, and the ER.
Yes,
right? And then the principle, and then seems the scientists and why they this is the nature, right? It's the nature of the why they bind the iron 2, not iron 3. This is the question, I think. And
for the discrimination of iron 2 from iron 3, we can explain because this is kind of reduction. So compared to Fe3 plus, Fe2 plus is more reductive because it has lower redox state. So that is why this can discriminate Fe2 plus from Fe3 plus.
They have a different. I think they have a different affiliation.
Not affinity, but redox.
Oh, they make a change. Yes. It's not the binding, but this is the interaction. So no.
No, chemical reaction.
Yes.
So, yeah.
Yeah, this is a reaction. So the answer is the probe make the iron 2 convert into the iron 3. It's not the directly binding.
Right.
Yeah, and then make
just contact.
The contact.
Not bind, but just contact.
Right. Yeah. Okay. I think once it, and then another one is I just read it. Most of studies have investigated the change in gene expression on iron on ferroptosis in presence of ferroptosis inducer. May I ask for your opinion on the regulation of the gene regulation on iron ferroptosis in the absence of inducer? So it's not really related to your talk.
Oh,
I think.
Right. I think so.
If so now I think now we investigated the ferroptosis. Most of the time we always use the ferroptosis inducer, and not really another way, right?
Yeah. Exactly. Thank you. Nice comment.
That's a comment. And do you have any comments on it?
Yeah.
And then thank you very much again and for exciting work. And then let's move to the third speaker.
Sorry, maybe we have to have a coffee break.
Okay. Oh, okay.
Yes, sorry. The next presentation will restart at 15:30.